Hello viewers, to continue with our discussion on the genic control of sex determination in plants, let me just enumerate another very interesting aspect of the lower plants, that is mosses, wherein the sex determination has other interesting ramifications. A recent study shows that the male and the female plants of the fire moss, Ceratodon purpureus, it emits different and complex volatile organic scents. Now, this must be thought of some sort of pheromones. Now, it is seen that the female plants are emitting more compounds than the male plants, and on your screens you can see a picture of the fire moss, that is a Ceratodon purpureus, with its conical sporophytes and still more sharp calyptra. So, it is seen that the hexapod insects, which are called as the springtails, they only choose female plants preferentially, and when they are choosing these plants for movement, they are enhancing moss fertilization. What do these results they seem to suggest? They suggest that there is a plant pollinator relationship which is quite analogous to those found in the flowering plants. So, that means the ingredients have evolved, and these studies have found that the fire moss chromosomes are much older than even the dinosaurs, and they have maintained a lot of genes, many of which have been evolving maleness or femaleness over time. So, why are we stressing on the moss genome all the time? Because the ability to look at purely male or female genes in these moss species may have very big implications on the plant world as a whole, because many of the genes on the moss sex chromosomes are known to control fertility in flowering plants and crop plants. So, the control of reproduction in crop plants is important for food production, and the moss genes may provide valuable new tools to boost female or male linked function. In this part of our discussion, what we now have to study is some more examples of genic control of sex determination in plants, but we shall begin with some very interesting information on entherogens and sex determination in ferns, which has been provided to us by modern molecular marker techniques. You remember, viewers, we had made a discussion on Ceratopteris fern, and this particular fern, we had mentioned that it was a homothallic situation, but then depending upon the presence of entherogens, it could have either of the fates. Either it could develop into a male thallus or into a female one. So, now a fund of new information has been accumulated, because Ceratopteris, as you know, has a haploid number of 37, and it has two genes which are called FEM1, and it has two genes more which is called the TRA genes. Now, together these FEM1 and TRA genes are promoting the differentiation of male and the female traits respectively, that is FEM for Entheridia and TRA for Archegonia. Sex chromosomes, mind you, have not been described for any ferns or lycophytes, and by the way, these two genes FEM and TRA, they are antagonists. So, that means if FEM1 is going to be active, TRA is not, and vice versa. Now, the question arises, which of the two genes prevails in the gametophyte? at a particular time, and thereby its sex will be determined by a pheromone, which is antheridogen and which is abbreviated as ACE, ACE. Now, this particular antheridogen is going to activate another gene which is called as HER, and there are at least 5 of these, and then it triggers a series of switches that ultimately result in male development. Viewers, you would be familiar with the idea that one of the genes only can be on and then the other would be off. So that means male development will take place only when FEM1 is on, which obviously means that its antagonist TRA is off. So, and when it comes to the origin of a female, then these switches will operate in the opposite direction, where the spores will germinate in the absence of ACE, because when ACE is there, then maleness will be there. So that means it's a case of repression of between FEM and the TRA. But then this repression is not direct. The relationship between FEM and TRA is through certain other regulators which are abbreviated as NOT1 and MAN1. So that means the TRA is going to activate MAN1, and which is going to repress FEM1, and FEM1 is going to activate NOT1, which represses TRA. So in other words, this NOT1 and TRA, NOT1 and MAN1 are considered to be the regulators of the regulators. So it's a complicated process. Let us see on our screens how the story goes. The idea is, if you can see on the left hand side of the diagram, ACE is present and it is written as plus. When ACE is present, that means the genes HER are activated, and when they are activated, then FEM is also there, and FEM is now responsible for the origin of a male gametophyte. On the right hand side of the diagram, now ACE is not present. If ACE is not present, then HER is also not activated, and then now TRA comes into picture. And when TRA comes into picture, that means the MAN is also there, and it would not allow the maleness to come, because and antheridogen is not present. So it would naturally give rise to a female gametophyte. The genic sex determination, as I said, is either by single gene or by multiple genes, and it occurs in both dioecious and monoecious plants. For example, in papaya, asparagus, cucumber, vitis, abelium, spinach, and many more. Let us take an example of Abelium elytrium, which is a member of Cucurbitaceae. I may just add, viewers, that Abelium along with Bryonia are two very important members of Cucurbitaceae, which constitute very important homeopathic medicines. They are extremely rich in cucurbitacins. So a single gene determines sex in this Abelium, which is commonly called as the squirting cucumber, and a single gene has 3 alleles. So it's a case of multiple alleles. Maleness is controlled by A capital D, femaleness by A small d, and the monoecious nature by A plus. The dominance pattern is A plus greater than A small d greater than A capital D. So naturally, any given variety is either monoecious or dioecious. On your screens, you will find a cross between a female which is A small d and a male which is A capital D and A small d. And finally, the progeny will be in the ratio of 1:1, that is 50% male and 50% female. Surprisingly, 99% of the A plus AD are monoecious. Actually 100% should have been. But then 1% of this genotype are female. Now what does this indicate? It indicates that perhaps some modified genes are also present, which are present at a locus other than A, which are modifying the situation in such a manner that all A plus A small d are not monoecious, only one are female. So if you cross a monoecious and a female plant, as you can see on your screens, you would again get a monoecious plant. Sex determination in maize also makes a very interesting example, because in Zea maize you will find that there is a diploid sporophytic stage which is predominating, and both the male and the female structures are present on the same adult plant. So what does it indicate? It indicates that sex determination must occur differently in different tissues of the same plant. It would be different at the apex and it would be different in the lateral meristems. As you can see on your screens, in this monoecious species, the male and the female flowers are present on the same plant. However, there are two types of inflorescences whose positions are also different. I may add, learners, that maize has a diploid number of 20. 2n is equal to 20. And there is a terminal inflorescence, as you saw in the diagram, which bears staminate florets. And it develops from a shoot, apical meristem, and we call it as a tassel. So tassel would be male. And the lateral inflorescence, which develops pistillate florets, it comes from the axillary bud. So we call it as a ear or cob. On your screens you can see a diagram of a maize plant with the tassel and the silk. The cob part is also called as silk because it has a silken structures. Now what is the genetics behind the whole aspect? A recessive gene small b small a controls the barren cob in a homozygous condition. That means small b a. Now the cobs, they become barren or they become non-functional. There is another gene small t small s. If it is present in a homozygous condition, it is going to, that is small t s, it is going to convert the male flowers of the tassel into female flowers. So this is an example of single gene sex determination. And if genes of both the barren cob and genes for tassel are in recessive form, that is the B and A and T and S, then the monoecious plant is converted into a dioecious plant, which is a female. So this is a case of selective abortion of stamens in the ear florets and pistils in the tassel florets, and this would account for unisexuality. So in other words, the difference between monoecious and dioecious is rare in maize. It depends on the substitution of two single gene pairs, as I said before, BA and TS. So the allele for barren plant in a homozygous condition, as I said, is going to make the whole things staminate. And the allele for tassel seed, it is going to transform the tassel into a pistillate structure. On your screens, learners, you will find a summary chart of what the different genetic constitutions in Zea maize would lead to either a rudimentary female, a male, a monoecious one, and a normal female, and also the modifications which are there due to the presence of either the double recessive presence or the recessive and dominant presence of the BA and TS genes, or double dominant and the dominant recessive of the other ones. Let us take another very interesting example of sex determination in papaya. As you know, learners, Carica papaya is a diploid polygamous species. It is a trioecious one because we see all three types of papayas: female, male, and hermaphrodites. And interestingly, the hermaphrodites are the sweetest of all, and basically, they are self-pollinated. And if you are engaged in the papaya cultivation, then it's very cumbersome process to remove the unwanted male plants, because the male plants are not going to have the fruits. So it's very difficult to remove these unwanted male plants from the dioecious papaya plantations, and it's very difficult to even recognize them earlier. So this will impede naturally large scale papaya cultivation. Now, what is the genetics behind it? A single gene with 3 allelic forms controls sex determination in papaya. M1 is the dominant factor for maleness, as you can see on your screens. M2 is a dominant factor for hermaphroditism, whereas small m is the recessive factor for femaleness. So it means if there is a single gene with 3 alleles, then if a genotype small m, that means homozygous, recessive, it would turn to be a female plant. If we have capital M1 and small m, then the male plants would be there. And if we have capital M2 and M, hermaphrodite plants would be there. And interestingly, the other genotypes where we have both the capital M's in any of the forms, whether it is M1M1, M1M2 or M2M2, they are inviable because they show this type of a dominant lethality. Let us see some of the crosses on the screen. For example, if we have the parent as small m, which is a female, crossed to a male, which is M1M, then the progeny are 50% male and 50% female. Likewise, if we have the cross between not M1M, but we have with M2M, then also we have the 50% male and the 50% female as the progeny. And if we were to self the genetic constitution of capital M small m, then also we would find that the gametes are such that we will have the M1, M2 in a dominant form, they would be inviable and only the heterozygous would remain. On your screens, learners, you will find the sex ratio and the summary of how the different genotypes of capital M1, capital M2 and small m are going to result in the sex ratio of the individuals. However, papaya, as we know, is a diploid, and the current understanding regarding the papaya sex determination is that it is also recognized in terms of sex chromosomes. There are 2 types of Y chromosomes which are slightly different. There is a genic balance between the sex chromosomes, and there are various genetic switches which are being governed even by hormones. We would have the opportunity to talk about these when we are talking about the ABC model of a floral differentiation. And therefore, in a monoecious species, developmental regulation would lead to the production of different genders, as we have seen that in maize and cucumber, several genes affect the gender of flowers. And if there is mutation in these genes, it would lead to either feminization or masculization of flowers, or to change even the position of the male and the female flowers on a plant. Let us then talk about the example of cucumber, because it also has different loci which are controlling sex. In Cucumis sativus, as it is botanically called, the sex is controlled by 2 loci, capital F and capital M. And it is an interaction between these two which is going to produce three different sexual phenotypes. On your screens you would find a summary chart that if at least one capital M and capital F is present, then a plant is gynoecious. If we have just one capital M, then it becomes monoecious. And if it is small m but only one or at least one capital F, then it is a hermaphrodite. And it would be andromonoecious when both the genes are present in a recessive form. So that means a third gene, there is another gene which is present with a locus, and this is going to intensify the production of male plants in condition of what we call as recessive homozygosity. So that means the genotypes having small a along with the capital M are definitely going to result in the origin of a male plant. And if the A itself is heterozygous, then the plant would be monoecious. There is another very important sex determination mechanism in the annual mercury plant, which is Mercurialis annua. It is a member of U4BAC. Learners, you must remember that the species of Mercurialis annua is capable of producing about 135 million pollen grains, which is enormous. And the sex is determined in this case by three genes, A, B1 and B2, which is going to make the species either monoecious or androdioecious. Now, male plants should have at least one dominant A and one dominant B. Now, this B is either B1 or B2, doesn't matter. The female plants must have recessive of any of these two genes, and it is seen that cytokinins are also very important in affecting the gender dimorphism. There are different types of males here, because there are weak males, they will have at least one allele of the dominant B2. There are strong males which have both capital B1 and capital B2, that is in a dominant form. There are intermediate males which have at least one allele, either B1 or B2, and there are weak males again which have at least one of the B1. So, weak males would be either capital B1 or capital B2. And on your screens you can see a summary chart of the different types of combinations, genotypic combinations, which may lead to weak, intermediate and strong males along with the females and the monoecious ones. So, viewers, with this little information, we will now pass on to our next lecture, which would be comprised of a detailed discussion on melandrium sex determination, the chromosomal sex determination of the plants, and along with the new information on the ABC model and the other genetic information. For this lecture, I have used the bibliography which is there on your screens, and some of the papers by Charles Worth and Milewitz and Sawatsky are very pertinent to get an exciting information on plant sex determination. Thank you.